Yesterday we learned till here, right? Nag, Nam. And one more thing I had to tell you was this. See, evolution, how it happened. First, Monera came. This was the first theory, okay, according to scientists. First, Monera came. And after that, Protista. And from there, Plantae, Fungi, and Animalia. Okay, now we will learn about first kingdom, that is our kingdom Monher. This kingdom, what does it contain? It contains bacteria, okay, bacteria, mycoplasma, and actinomyces and cyanobacteria, okay, archibacteria, okay, under the five kingdom classification. And if we are talking about sixth classification, archibacteria will belong to another kingdom. Okay, now let's see this general characteristic of this Monera. It is unicellular. Okay, it is unicellular prokaryote. That's the main thing. Okay, and first, who came first? Which was the living organism? First living organism? It was Moner. Okay, most ancient, most primitive, and they occur in various places, extreme places also. Okay, anywhere you will find bacteria. Okay, you will find them in snow, you will find them in hot springs, you will find them in lava, desert, deep ocean also. You will find bacteria. Okay, anywhere, everywhere you will find soil, everywhere you will find bacteria. In if you pick a mouthful of sand, what will happen? You will find there is millions of bacteria in. So bacteria is unicellular prokaryotes, okay, and first inhabitants, that means first living organism on the earth were bacteria. Then it is most ancient, okay, most old and primitive. Their cells is primitive, okay, and they occur everywhere. That's why it is called cosmopolitan. And they are also found in extreme habitats, okay. Hot spring, right? What happens in their temperature is here 200 degrees Celsius. If we were there, what would happen to us? Will we survive?
We'll burn.
Yeah, we won't. But bacteria live there. And in desert also, where the temperature is high, we do live. But the thing is, we don't live particularly hot desert. Okay, and the snow also, in snow, right? We play, but we don't live in the snow where all the time, in the mountains where all the time there will be snow. We don't live there. We just go for touring or something like that. We can't live for longer, right? But the bacteria, they can live. And the deep ocean also, scuba diving, all that we can do, but we are not able to live there. Okay, there the pressure is high. In the snow, pressure is low. Okay, the bacteria, right? Those bacteria which live in extreme environment condition, those bacteria are called eubacteria. Okay, and the bacteria, we are talking about bacteria. What is the mode of nutrition? Can you tell me? In the previous class, I have discussed mode of nutrition of bacteria, Monera. Ria, tell me.
Ma'am, can you repeat the question?
Okay, mode of nutrition of Monera, kingdom Monera.
Ma'am, I don't know.
How do bacteria get their nutrition? Can you guess? You have heard of bacteria, right?
Yes, ma'am.
Yeah. Then what else have you heard about?
Ma'am, they can live anywhere in any
Yeah, that-
temperatures.
And how do they get their nutrition? They are both
Other
autotrophic and heterotrophic. Okay, autotrophic. Some of the bacteria are photoautotrophic, like blue-green algae. That is bacteria. Okay, and then we have chemotrophic bacteria. Purple bacteria. They sulfur purple bacteria. What do? They get their energy. Okay, they prepare their food from chemical energy. Okay, and the bacteria, blue green bacteria, how do they make their food? They make it by photosynthesis. Okay. Next, also the bacteria are autotrophic and also heterotrophic. Okay. So heterotrophic, what would be the mode of nutrition? What kind of heterotrophic? They could be parasitic or they could be saprophytic. Okay. In parasitic, if they are living inside the body of a host, okay, and that will be known as what we have learned in 10th. Ria, tell me, if parasite is living inside the host, getting nutrition from the host while living inside, what type of parasites are they called? In. They are known as endoparasite. What about out? That means
Ecto.
living in surface. What did you tell, Ria?
No, I don't know.
Ectoparasite. Got it? So endo and ecto. And also how they do respiration. Okay. What kind of respiration? Two kinds. Basically, there are two kinds only. What are the kinds? Types of respiration. LFP chapter in the respiration we had learned, right?
Yes, ma'am.
Two kinds of respiration.
Anaerobic, aerobic,
Yes, good. Anaerobic and
aerobic respiration.
okay. So those bacteria which respire in presence, right? They do their respiration in presence of oxygen. That is known as aerobic respiration. And in absence of oxygen, anaerobic. Okay. Now, see, I have already told you most of the bacteria are heterotrophs. This is given in your NCERT book. Okay. And some are autotrophs. Okay. This is the sentence. Now, if the question is given like this, statement number 1, 2, and 3. In first statement, it is given that some of the bacteria are heterotroph. In the second statement, we have few of the bacteria are heterotroph. And third statement is given, most of the bacteria are heterotroph. Now tell me, in these statements, which is the correct one? Okay. In your book, right, it is given most of the bacteria are heterotroph and some are autotrophs. But in the first statement, it is given some of the bacteria are heterotroph, right? Some it is given, but in your book, it is given most. So the statement number 1, is it correct or not? Ria, hello.
Ma'am, I couldn't hear you for 5 minutes. 2 minutes, I couldn't hear you.
Am I clear? Hello.
Yeah, now you're audible, ma'am.
Okay. So in your book, right, in NCERT book, it is given most of bacteria are heterotroph and some are autotroph. If
Yes,
the
ma'am.
question is like this, statement number 1, 2, and 3. In that first statement is given some of the bacteria are heterotroph. Second is given few of the bacteria are heterotroph. And third statement is given most of the bacteria are heterotrophs. So which in these three statement, which is the correct?
Ma'am,
How
third,
will you do it? Huh?
third statement?
Yeah, third statement, because it is given most of the bacteria are heterotroph. Why not first and second?
Ma'am, because most is majority of them. Some would not be majority and few would be lesser than majority. So it wouldn't be the right answer.
Good. If here few of the bacteria are autotrophs, if that statement is given, then you will tell, yeah, this second statement is good, right? Correct. Okay.
Yes, ma'am.
So this is done. And about this bacteria, what do they have? What kind of cell do they have? First, they will have cell wall, right?
Yes, ma'am.
And what is it made up of? We have discussed it so many times. What is it made up of? Cell wall of bacteria.
Ma'am,
Peptidoglycan.
Okay.
Peptidoglycan. So this peptidoglycan, peptide,
Yes, ma'am.
that is our protein. Okay, protein meaning amino acid, peptides. So many amino acids, right, they form peptides. Okay, and that peptide will form protein. So peptidoglycan. So the content is what? Peptide that means protein. Peptidoglycan. What is that glycan? Glycan, sugar, glucose, glycan. Okay. So
Okay, ma'am.
sugar. So this you can say simply it is made up of this protein and sugar. Okay.
Okay, ma'am.
Now, which type of sugar? It is NAG and NAM. NAG meaning N-acetyl glucosamine, okay, and NAM meaning N-acetyl muramic acid. In the Archaebacteria, what will be not present? We have seen in the last class. This NAM will be not present, okay? NAM will be present. NAM will be not present in Archaebacteria. That is the only difference between the cell wall, okay?
Yes, ma'am. Can I copy the name?
Yes. Actually, I will be sending PDF, but my it is not syncing. Okay,
Okay.
I'll send you PDF also.
Thank you, ma'am.
Mm.
I'm done.
Done.
Yes, ma'am.
So this is what I have told you. Peptidoglycan, peptide, that is protein. Okay, and this peptide is made up of amino acid, and this glycan, what is that? It is sugar. And which type of sugar? NAG and NAM.
Okay.
And this peptidoglycan, it is also known as murin and mucopeptide. Okay.
Okay, ma'am.
Cell wall of the bacteria is non-cellulosic, right? It does not contain cellulose. It contains peptidoglycan. We had this, then we have given cell wall, okay? Cell wall, I have told you, peptidoglycan, polysaccharide and amino acid, right? Sugar, you can say sugar as saccharide, okay? Like in the protein, you say peptides. Many of the saccharides, they make sugar, starch, okay? Which is the kind of mucopeptide. Cell membrane, okay, here also the cell membrane is made up of lipoprotein, like in the eukaryotes. And the space between the cell wall and cell membrane, it is known as periplasmic space. So, the space between this cell wall and the cell membrane, what is known as what is it known as? Why is that spelling? Plasmic. Okay. And next, what do we have? Next, cytoplasm. We will get into the cytoplasm. In this cytoplasm, right? This fluid substance.
Ma'am. Ma'am, sorry to interrupt. There's someone at the door. Can I go open the door and come?
Yeah.
Thank you.
No, this is correct. You are back.
Ma'am, I'm back.
Okay. So next we'll see cytoplasm, right? In the cytoplasm, what do we have? Usually in the eukaryotes, we had cell organelle, right? Membrane bound cell organelle. But in here, we don't have any cell organelles, membrane bound cell organelles. The only organelle which will be present here is ribosome. It is non-membranous, okay, and also mesosome. This is the structure, but here it is not given. Next, you can see what will be not present. Mitochondria, will it be present? Is it membrane bound organelle?
Yes, ma'am.
Yes, it is membrane bound organelle. And how many layers are present in them? Mitochondria, double membrane.
Okay, ma'am.
Right? We have learned it already. Double membrane. Is it present in bacteria? No, because already it is given. Cell membrane bound organelles are not present. What about ER? Endoplasmic reticulum. How many membrane does it have? Single or double?
Double.
How many membrane does ER have?
Ma'am, I don't know. Double?
No, single.
Oh.
Golgi bodies, single. Nucleus, double. Nuclear membrane, it is double, okay? Nucleus has nuclear membrane. And ER, is it present in bacteria? No. Golgi bodies? No. Nucleus? No. Nuclear membrane? No. What about lysosome? Will it be present? It is also single. No. not present. Then what else is there? Mitochondria, wait, I have already given. Yeah, chloroplast. It is double. Will it be present? No. Then what do I mean by double? One membrane, second membrane. Okay, that is double. Then lysosome. One more thing here. Any other? No. Lastly, ribosome. Ribosome are present. Okay, in the bacteria, ribosome are present. And which type of ribosome? 70S ribosome. This S is sedimentation coefficient. Okay, this S is sedimentation coefficient. And this is unit. Okay, Svedberg unit. This measures the density. Okay, density of the ribosome, size and density. So this one, this 50S and 30S, they form what? 70S. And ribosome unit, this S, right? It is not this unit. It is not additive. That means we cannot add 30 plus 50. 30 plus 50, what is that? How much will we get? 80,
80.
right? But the unit is what? 70. Okay, so 30 plus 50 we cannot add. This is not additive unit. Okay, this 70S is made up of 30S and 50S. Okay, see, this is the prokaryotic ribosome. Here we have the 30S unit, and here we have the 50S unit, and 30S unit we have 16S RNA. What type of RNA? These are all the different types of RNA. And in 30S we have 16S RNA, and in the 50S RNA we have 23S RNA and 5S RNA. Okay, this S. And here in the eukaryotes, what do we have? 40 and 60. Larger subunit is 60 subunit, and the smaller subunit is 40 subunit. Okay, so in the eukaryotes we have 80S ribosome. In the prokaryote, which type of ribosome we have? 70S ribosome. What does ribosome do? It helps in synthesis of protein. Okay, and ribosome is non-membranous. It doesn't have any membrane. That's why this is present in bacteria. Okay, because it is given membrane bound cell organelles are not present. All others are membrane bound. Only the ribosome are non-membrane bound. Okay, and we have seen here. Next, what do we have? We have seen all the cell organelles. Then we have the nucleus. Nucleus is not present. So what is present? Genetic material is present, and it is known as nucleoid or xenophore. Okay, we have circular DNA, RNA, and polyamine. In the humans, eukaryotes, what did we have? We had histone protein. What was its work? It helped in the packaging of the DNA. Okay, histone protein. It was basic, positive, and the DNA is negative. That's why it could get folded, and it will form what? Get condensed, and it will form what? Chromosome. Here we don't have histone protein. Instead of that, we have polyamine, which is used for the packing. Okay, not fully packed, but yeah, somewhat packing. That's why it is known as naked DNA. Not only because it has no covering, that is, nuclear membrane is not present, also that, and also because it doesn't have histone. It doesn't have histone protein. Got it? Got it or not?
Yes, ma'am. Got it.
So this bacteria, right? Bacteria is simple in organization. That means its structure, right? Its cells is very simple. What we have? Only the cell wall, right? And we have the cell membrane, right? And after that, we have the ribosome, and we had one more thing which was known as mesosome. Okay, infolding, right? There will be particularly what infolding. Okay, and that right here we will have what mesosome. So this mesosome, right? It helps in cellular respiration because it will contain respiratory enzyme because there is no mitochondria. So the bacteria, it needs energy. So from where would it get energy? So there will be mesosome, and in that we will have respiratory enzymes which will help in producing the energy. Okay,
Okay.
and there would be also one more thing, plasmid. What is that? It is extra chromosomal DNA, extra DNA. Okay. Nucleoid will be present. Okay. But and also one more extra DNA will be present. Okay. Circular DNA, that is known as plasmid. Okay. It helps in the reproduction, and it helps in the resistance. Okay. Resistance against the other bacteria. Okay, it codes for resistance, this DNA, this plasmids. And these plasmids are very useful in biotechnology, which you will learn in 12th class. Okay, we have learned everything in this bacteria, and it has this flagella. It will help in locomotion. Then it will have capsule for protection, and also it has pili. Okay, pili is for reproduction. It is then yeah, where were we? Simple in organization. So it is very simple, right? There is no tissues, no organ system, right? All those complex things are not there. Not many mechanism, right? Metabolism, reactions, all that things are not there. So it is very simple, very simple in organization, but its behavior is very complex, right? Behavior is complex. How it is complex? When we have fever, or like cholera, that time we'll have vomiting because of the bad water, right? Because it has the bacteria which will, if we drink the unclean water, the bacteria will get inside us, right? And also there is all these bacterias, right? If we are not taking our medicine fully, like the doctor will give you three course or four course, and they will tell you to take all the course, complete it fully. Even if you feel better, you have to complete it fully. Okay, why? Because, right, the bacteria, some of the bacteria, if we are taking the medicine only for 2 days, what will happen? The bacteria are not totally dead, right? They would be in the they would, some of the bacteria will be alive, right? All the bacteria will not die. So what will happen if we are not eating? Again we'll get infection, again we'll have fever, right?
They'll regrow again.
Yeah, they'll regrow. So the behavior of bacteria, it is very complex. Some of the bacteria, they will behave like the other bacteria. They will change their shape, and some of the bacteria will behave like they are like us, ours, like our cell, so will not be recognized. Our cell, our immune system will not be able to recognize them. So those are very complex, right? Behavior is very complex, but it is very simple structure. Got it? So if the question is given, complex in organization, simple in behavior, is this statement correct?
Yes, ma'am.
Yes. See that statement again and tell me. Can you see?
Yes, ma'am.
So tell me, is this statement correct or not?
Ma'am, it's not because they're not simple in behavior.
Yeah, see, they have changed the only one word, interchanged. The simple should be coming here and the complex should be coming here, right? Only
Yes,
those two changes. And because of that, the whole meaning is different and the statement would be wrong. That's why when you read the statement, you have to be very careful. Okay, statement based questions are, yeah, this is how they will give you. Okay.
Okay, ma'am.
Next, what we have? Yeah, shapes and sizes. Based on the shape, right? There are 4 types of bacteria. We have Bacillus, Coccus, Vibrio, and Spirilla. Okay, Bacillus, their shape would be like this. Okay, rod shape. Rectangular, cylinder, okay, cylinder shape. And this Coccus will have spherical shape. And Vibrio, they will have which kind of shape? Comma. And this Spirilla, we will have shape spiral. Okay, you can find this here. This is cocci. Why I am telling cocci? Because there are many coccus. 1 coccus, 2 coccus, 3 coccus, 4 coccus. So there are many coccus. That's why cocci. Plural of coccus is cocci. And here we have bacillus. This bacillus are what? Rod shape. And
And they look like a spoon.
yeah, here rod shape, right? So this bacterias, if there are many of the bacillus, then it is bacilli, and this is spirilla, plural, and a single, it is spirillum. Okay.
Yes, ma'am.
Next, lastly, we have comma shaped bacteria. It is known as plural form Vibrio. Only single, okay, single. It is Vibram. And here example is also written. Cocci example, this Coccus bacteria example, Diplococcus pneumoniae, the bacteria which cause pneumonia. And then we have E. coli and Bacillus anthrax. Anthracis. This bacteria causes anthrax. It is bacillus. Which type of bacteria? It is bacillus. And then we have this spirillum. Example is Spirillum voluntas, Streptonema. In this bacteria, right? One example, if you remember, it is enough. Like for the coccus, we have example Diplococcus pneumonia. Okay, and here we have example E. coli or Bacillus anthracis. And here we have Treponema. And the Vibram, we have Vibrio cholera, the bacteria which causes cholera. Okay, so these are the types of bacteria, and here we will learn if the cocci spiral, okay, if there is only one, if they are occurring single cell, they are not occurring together, then it is known as monococcus. And if they are occurring in pair like this, one coccus, the other coccus, this is known as diplococcus. Okay,
Yes, ma'am.
here it is written as tetracoccus. That is wrong. Okay, this is tetra means 4. Okay, diplo means 2. In pairs they appear. Then in group of 4, if it is occurring like this, all, how many bacteria are present in here? Can you tell me?
And 4.
Yes, 4 coccus. And you can tell cocci. So here it is known as tetracoccus. And if it is occurring like in the chain, in the form of chain, okay, and that will be known as streptococcus. And if it is occurring like this, like sheet or like grapes, okay, this is known as Staphylococcus. Got it? Ria, got it?
Yes, ma'am.
Based on the shape, based on how they are arranged, right? The name will be different. And the bacteria, this bacillus also, it's like same thing. Monobacillus, Diplobacillus, Streptobacillus, and here only the name change here is this one for the layers, right? For the sheets. In the case of coccus, it was Staphylococcus, but here it is Palisade Bacillus. That is the only difference. All the other naming are same. Spiral, only spirillum. And also other shape is also found. Mycelial and stalk. We have done. They are found in all kinds of habitat. Okay. Unicellular prokaryotic organism, okay. Then contain peptidoglycan, okay. Have extracellular chromosomal DNA segment called plastid, already done, okay. Cell organelle absent, okay. All this is done. Cytoplasm, there is no true chromosome. False chromosome, what is that? It is made up of circular DNA, non-histone protein like polyamide. Okay? Ribosome 70S type for the protein synthesis. Example, eubacteria, blue-green algae, archaebacteria, and mycoplasma. Got it? Everything I have taken here, right? Anything missing? No. Motility, based on how many flagella is there, right? Bacteria are named or categorized into different types. So A-trichus meaning no flagella. When flagella are absent, right, in bacteria, it is known as A-trichus. Okay, and example of that bacteria is Micrococcus and Pastorella. Here, both the bacteria, they don't have any flagella. Flagella is absent, and they are known as A-trichus. Monotrichus. If the bacteria is having a single flagella, okay, that bacteria are known as monotrichus. Vibrio and Thiobacillus and Pseudomonas, they all have single flagella. Then amphitrichus, the bacteria, right, they have flagella on both sides, okay. Nitrosomonas is the example. Next, Lophotrichus, here bunch of flagella are present, okay, like this. And Cephalotrichus, here bunch of flagella are present only at one end. Example, Corneobacterium. Here, Locriticus example is Salmonella. Salmonella typhi, the bacteria which cause typhoid. Then Peritrichus, here whole body of the bacteria. What is present? Flagella.
Ma'am, I have to go for another class. I have test today.
Okay. Yeah, 5 minutes, right? Only this is last. Here example again we have E. coli and Salmonella typhi, okay. Here also we had the Salmonella, and here also we have the Salmonella typhi, okay. Few of the species will have this and the other will have this. Got it? So 6 forms. Based on the motility, the bacteria are categorized into 6 forms. Okay, 6 types. Any doubt?
No, ma'am.
Okay, then bye bye.
Thank
See
you,
you in
ma'am.
next class.
Thank you, ma'am. Bye, ma'am.